Hi everyone, welcome to the live stream. Also from now on, the live streams will be from 12 to 1 because I got a couple of requests. So from this week on, this will be the regular time for the live streams. All right, since it's 12, I'm gonna go ahead and get started. Pull out the worksheet if you have access to it. So this week we're going to be looking at neuron function and what type of things can affect it and in what ways. So the first question says, some nerve gas and insect poisons work by destroying acetylcholine esterase. Acetylcholine esterase is normally present in acetylcholine synapses and acts to degrade acetylcholine. What is likely to happen to nervous transmission in insects exposed to this insect poison? All right, does anyone have any guess on what will happen if the insect is exposed to this poison? I'm not sure when this comment came in, but it says, is this the usual time for the meeting will take place from now on? Yes, this will be the normal time from now on, from 12 to 1 on Thursdays. All right, so in this question, they're talking about acetylcholine and a neural toxic product that's going to affect that. So first we need to know like what acetylcholine is, right? So in the central nervous system, acetylcholine can cause inhibition or excitation of the postsynaptic neuron, but like which one it is going to depend on the receptor. So how a neurotransmitter acts on a neuron is dependent on the type of receptor itself. But we know in the neuromuscular junction, like voluntary muscles, like when we move our arms and stuff, acetylcholine is going to be excitatory in those types of muscles. And we also know in the heart muscle, the effect of acetylcholine is going to actually reduce the rate. And so this question is saying that acetylcholine esterase is being destroyed. And we know acetylcholine esterase removes acetylcholine or breaks down acetylcholine from the synapse, right? So if cholinesterase is destroyed, then acetylcholine will remain longer in the synapse. And if it remains longer in the synapse, then the postsynaptic neuron will be continuously stimulated by the acetylcholine. And that'll either depolarize it if it's excitatory. So, like in our muscles, that will continue our muscles to keep contracting, but then it can also hyperpolarize if the receptors are inhibitory. So wherever acetylcholine is inhibitory, this neurotoxin is going to hyperpolarize, and where acetylcholine is excitatory, the neurotoxin is going to keep it in an excited state. Does that make sense? I know that was a lot of words, so if you have any questions, please let me know. All right, so in the next question, it says pufferfish contains poison tetrodotoxin. In some shellfish, produce a paralytic poison called saxitoxin. Both of these poisons block the sodium channels in neurons. What specific effect could these toxins have on the neuron functions? All right, what do you guys think will happen if the sodium channels are blocked in a neuron? Do you think action potential will generate? Action potential won't generate? Yeah, I see a response that says won't, and that's the right answer. So we need the sodium channels to be open in order for the cell to generate an action potential. So if these toxins block that, then the axon will be unable to generate an action potential. Great job. All right, next we're looking at a type of spider, and that produces a toxin that blocks the calcium channels. So unlike the sodium channels, we don't need the calcium channels for an action potential to occur. Calcium helps with the with neurotransmitter release. So in this case, if the calcium channels are blocked, we will still be able to produce an action potential. All right, I know I kind of talked about this just now, but what do you guys think will happen um, in a signal across synapse using neurotransmitter if the calcium channels are blocked? Do you think neurotransmitter release will be affected or won't be affected? So for this one, we're going to need an influx of calcium in order to release our vesicles to fuse with the membrane. So if the calcium channels are blocked, no neurotransmitters are going to be released into the synapse. So no signal will be transmitted across the synapse. So we'll still have an action potential, but it just won't be able to go from one synapse to the other one or across the synapse. All right, for the next couple of questions, we're going to have to keep in mind the information given in the chart. And we need to keep in mind that resting potential of a membrane is negative 70 millivolts, and it needs to reach, or it reaches positive 55 when it's stimulated. So the first question is saying that we change the solution bathing the neuron by increasing the potassium concentration to 40 millimolar, and it's asking what effect it's going to have on the neuron. So we need to keep in mind that the axon permeability to potassium ion is much greater than to the chloride ion, which is much greater than the sodium ion, right? And at rest, the concentration of negative ions inside and outside are approximately equal, and the equilibrium potential for each ion is dependent both on concentration and electrical gradient. So it's not one or the other, it's going to be both. So so what so when potassium ions cannot be added without simultaneously adding a counterbalance anion. So for example, chloride. And as a result, like adding potassium chloride is going to add both potassium and chloride outside of the membrane, and it's going to disrupt the equilibrium potential. And since the axon is more permeable to the potassium ion will quickly move into the axon to establish an electrical equilibrium gradient. So I'm just going to write that down. So if we do that, potassium are going to move into the axon. And this movement of the potassium into the axon is going to depolarize the axon. And for the next question, it's saying instead of adding potassium to outside, we're adding more sodium bathing fluid to the neuron. And as like we've talked about before, the sodium ion cannot be added without a counterbalancing ion. So we would need to add something like sodium and chloride. So before, the axon is more permeable to chloride, and the ions would move into and hyperpolarize the cell. So, but we know in both like parts, like question A and B, we have the sodium potassium pump, which is going to restore the sodium potassium gradient anyway. So, whenever there is a disruption to the equilibrium, the sodium potassium pump will establish that equilibrium. But for this question, we can write that ions will move into and hyperpolarize the cell. All right, any questions so far? All right, so next we're going to be looking at how the nervous system is organized. I'll just read this out because it's useful information. So neurons are single cells composed of processes called dendrites that carry information to cell body and axon that carries information away from the cell body. In simple terms, nerves are bundles of dendrites or axons or mixed axons and dendrites. Ganglions are collection of cell bodies, and brains are collections of ganglia. Processing and integration of information occurs as a result of interaction within and between the ganglion. All right, so in order to respond to a stimuli, at the least, we need a reception of the stimuli and then an action potential in the sensory neuron, and the central nervous system, which is going to do all the integration and which is going to generate the action potential. And that action potential is going to be generated in the motor neuron, which then is going to cause us to do something, whether that's like a muscle action or a gland. So basically we have a stimuli that goes in our sensory tract and then in the brain that produces an active potential in the motor, and then the motor is going to elicit the response that we need. All right, so in the space, I'm going to try to draw this system. So where they're talking about the minimum requirement, and hopefully that visual helps a little bit. So let's see. Let's say this is the central nervous system. So this is going to be our spinal cord. And then this is our gray matter. And then this is going to be our white matter. So here we're going to have this type of interneuron looking thing. All right, so let's say we have our skin cell here. There's a hair. And this is where we're going to experience this stimulus. And the axons here are going to carry the signal like that. And then here in this side, we're going to get our motor response. And let's say this is our muscle. And it's going to go integrate here. So this is going to be our sensory, and it's going in this direction. And then this is going to be our motor, and it's going to be in this direction. All right, next we're going to be looking at what type of different combinations need to be added to elicit different types of responses. So this question says the blink of an eye response is one type of simple reflex that occurs, for example, in response to water splashing out of a sink or a snowball aimed at your face. When this occurs, you blink, and only afterwards do you realize when you blinked. So here they're talking about like instinctual things, like you touch something hot, you like move your hand pretty quickly without even thinking about it, right? So our reflexes. And they're asking what we would need to add to the diagram to realize when you've blinked. So for any reflex, we don't need an inner neuron, but if we need to realize that we've blinked, we would need to add an inner neuron or another type of synapse to the sensory axon, and that would allow us to carry information to the brain to actually realize what has happened. So here we would need an inner neuron. All right, and then the next question is why does it take longer to realize why you've blinked than it takes for the blinking reaction to occur? So just blinking without thinking about it, that's going to be your simple reflex. Your simple reflex, like it says in the name, is pretty simple. It doesn't have an inner neuron, which allows for the reaction to occur a lot faster. Whereas when you're realizing it requires a process of thinking, right? And that's going to require inner neurons and it's going to take a lot longer. And this also makes sense because you don't want a reflex to take a long time because a lot of times it's there to protect us from harmful things. So if our reflexes took a lot longer to occur, then we might end up like hurting ourselves. right, so the next is we're going to look at what composes the central nervous system versus the peripheral nervous system. So the central nervous system is going to be the integration and processing center. And then the peripheral nervous system is going to have sensory neurons. And this is essentially going to carry information to the central nervous system and carry information from the central nervous system, again, to our effector muscles or glands. All right, next we'll be looking at another subdivision. So within the autonomic nervous system, we have two divisions, the sympathetic and the parasympathetic. How I like to think of this is for the sympathetic, I like to think of fight or flight. And then parasympathetic, I like to think of rest and digest. I say this because in the sympathetic, this is where we're going to have like upregulation. Like this is essentially the part of your nervous system that responds when you're in danger. So this is going to upregulate glycogen turning into glucose. And that makes sense because you need more energy, right? Whether you're like running away from something or you're in danger, you're going to need more energy. And then this is also going to cause secretion of epinephrine, and that's going to increase your heart rate because you need more blood pumping. And the blood flow is going to be switched. So instead of it going to like your stomach and other parts that you don't really need, it's going to flow towards your skeletal muscle in your brain. Okay, so the next one, the parasympathetic, like I said, I like to think of this as the rest and digest because this is what's activated when you're kind of relaxed after a meal, you're not in stress, you're not in any danger, basically like calming activities. And here we're going to have reduced heart rate, low blood pressure, increased digestion, and more glycogen production, so turning glucose back into glycogen. The one exception to this is the control of the reproductive organ, where the sympathetic and parasympathetic complement each other, whereas usually they kind of go against each other. So when sympathetic is activated, parasympathetic isn't, and when parasympathetic is activated, the sympathetic isn't. But the one exception to that would be the reproductive system. So here I'm going to write low blood pressure, increased digestion, increased glycogen production. Okay. All right, any questions so far on the divisions or other things that we've looked at so far? All right, let's look at the next couple of questions. All right, so Fibromyalgia is a condition that affects about 2% of the US population. Its primary symptoms are chronic pain, difficulty sleeping, and fatigue that is not relieved following sleep. Other reported symptoms include jaw clenching at night, dryness of mouth and eyes, an increased need to urinate, irritable bowels, muscle numbness, and tingling and headaches. Several researchers have proposed that fibromyalgia results from failed regulation of part of the autonomic nervous system. If this is true, failure of regulation of which division of autonomic system could account for these symptoms? All right, so for this one, they're asking if the autonomic nervous system, like which is it, the sympathetic or the parasympathetic failure that would cause all of these symptoms? And I would say, like, given the symptoms alone, we could say that the hyperactivity of parasympathetic system could be caused, or like one cause of this disease, and it accounts for all of the symptoms with the exception of dry mouth and muscle problems. Because we can see that there's difficulty sleeping, there's fatigue, irritable bowel, increased urination, and like increased urination and things like that would happen if there's a hyperactivity of the parasympathetic nervous system. All right, so to test their ideas, researchers recorded heart rates of both control and individuals with fibromyalgia for a 24-hour period of normal activity. They observed that patients with fibromyalgia showed greatly elevated heart rates, both while active and while asleep. So, with this additional information, it actually points us towards hyperactivity of the sympathetic nervous system, because earlier we looked at how the sympathetic nervous system increases heart rate. So, this could also be one reason. So, this specific symptom of higher increased heart rate could be a result of hyperactivity of the sympathetic nervous system. All right, any questions on the nervous system so far? Okay, next we're going to be moving into looking at sarcomeres and muscle functions and how they're organized. So the first question is, how are sarcomeres arranged within the muscle? So for this one, it's going to be in a linear fashion. So linearly microfibrils, and microfibrils are linearly arranged into muscle fibers. So it's kind of a hierarchy. So you have a sarcomere, and a bunch of them are going to make myofibrils, and then a bunch of those are going to make a muscle fiber, and then a bunch of those muscle fibers are going to make muscles. So I'm just going to do like an arrow diagram. So we have sarcomeres, and a bunch of them are going to result in myofibrils. And a bunch of those are going to result in muscle fiber, and a bunch of that is going to be our muscle. And all of these are linearly arranged. All right, so the next question is going to be about the sliding filament theory. So there's going to be a lot of content here. I'm going to try my best to be clear as possible, but you can also refer to figure 45.2 and 45.6 on your textbook for more of a visual. Okay, so essentially we have in the sarcomere, we're going to have myosin filaments that lie between thin actin filaments. And those are going to be attached to the Z line of the sarcomere. I like to think of the Z line as like the end lines that you would see of a sarcomere. And I remember that because Z is the last letter on the alphabet, so I always know it's like at the end. So the thick filaments are the ones that are going to have the myosin head. So thick filament equals myosin. And then, so so the thick filaments are going to have the myosin, and then they're going to alternate between a low and high energy state. In the low energy state, the myosin is going to be bound to ATP molecule, and then the ATP is going to be hydrolyzed to generate the myosin's high energy state. And the actin filaments are going to be combined of two strands of actin, and they're kind of in a helix form, so they're like twisted on each other. And the long strand of tropomyosin are also wound around that. And at rest, tropomyosin strands block myosin from binding to actin, and action potential in the sarcoplasmic reticulum and T tubules cause the release of calcium. And then the calcium is then going to bind on the troponin, which is on the tropomyosin, and that's going to change their shape. And once the shape changes, the myosin can bind to actin. And so essentially what's happening is you have your actin and your myosin, and they need to bind to each other in order to slide, right? But the actin filament has this like cover on top of it, which is going to block myosin from binding to itself, and that's called tropomyosin. But that can be released, so that block can be released when calcium binds to troponin. So calcium is released into this from the sarcoplasmic reticulum, and when that binds, it physically shifts a little and gives myosin the binding spot on the actin, and that's how actin binds, and you get the sliding filament theory. Okay, any questions on the sliding filament theory so far? I also like to watch like YouTube videos on the sliding filament theory because the visualization really helps. Like sometimes it's just like a lot of words, like when I'm talking or like when you're reading the book, but watching a YouTube video and just typing like sliding filament theory, it's just super helpful because they show like how the myosin actually binds and how tropomyosin actually moves out of the way. So I would highly recommend that. All right, so the next question is asking, how do your answers to question 1 and 2 helps explain why at maximum contraction a muscle measures 75 of its extended uncontrolled length? So this is asking why our muscle is shorter when it's contracted versus when it's not contracted, right? So the answer to this is because during contraction, the actin filaments are pulled towards the center of the sarcomere. So it's physically pulling. And at maximum contraction, the sarcomere's width is going to equal to the length of the thick filament. So So it's not that the thick or the thin filament is changing shape, it's that the sarcomere itself is just squeezing. So the thick and the thin filaments still stay the same, but everything else is getting squished. So that's why our lines are going to be different. I'm going to write during contraction. All right, so next we're going to be looking at neurotransmitters and how it helps muscle contraction. So acetylcholine is going to be excitatory in the neuromuscular junction, and this can release a trigger of an action potential along the length of the muscle membrane. And we're going to be describing the process. So in a motor neuron synapse within a muscle fiber, that's going to be in the neuromuscular junction. And if we get an excitatory action potential, then we're going to release acetylcholine. And what this is going to do is it's going to trigger an action potential in the muscle cell, and it's going to travel into the T tubules and release calcium. And like we learned earlier, calcium is going to help with the sliding filament theory. So basically what acetylcholine is doing is it's helping the release of calcium. So acetylcholine triggers action potential, and this action potential travels into T tubules, and that will cause the release of calcium. All right, any questions so far? All right, let's look at the next couple of questions. So the next one is, if acetylcholine always produces an excitatory response in the neuromuscular junction, how can we regulate within muscles are contracted and which are extended at a given time? For example, how can we bend the arm only partially? And so this is asking why we're able to control our arm in a certain way if it's always going to be excitatory, right? And this is because muscles are arranged into motor units, and the contraction of each motor unit is controlled by separate neurons. So we can control which motor unit is actually getting activated. All right, and our last question for the day, it's talking about severe calcium deficiency can lead to a reduction in bone mass. It can also have serious effects on the functioning of the nervous system and on the action of muscles. And explain what role calcium plays in the activity of muscles. So, like we talked about before, the release of calcium from the sarcoplasmic reticulum is ultimately what allows for the myosin to be able to bind to the actin. So, it's ultimately what causes the muscle to contract. So, if we have severe calcium deficiency, then muscles can become weak and it can even make someone incapable of a strong contraction. Oh, sorry, just saw these comments. So, yeah, I see a comment that says calcium plays so many roles. Yeah, I agree. Like earlier, we looked at calcium helping with neurotransmitter release, and then within a muscle neuron, we're seeing calcium helping with the muscle contraction. Yeah, it's definitely a very important mineral. All right, so that was our last question for today. Any questions on anything that we've went over or anything related to it? Okay, I'm gonna go ahead and put the extra credit link. Um, Make sure to fill it out so you can get extra credit. Also, in the extra credit form, if this time doesn't work for you as much as it did the other time, please let me know because I know last time people were requesting Thursday times, but we got like a lower turnout than we usually do on Tuesday. So if it's a worse date, then please let me know. Okay, I see that you like the earlier one. Okay, yeah, I'm gonna see maybe like in the form mention it because I'm gonna see how many people like the earlier one versus the one on Thursdays. I know a lot of people also wanted it later because they wanted to go over it in class before we went over it, but like considering how many people showed up today versus the other ones, I'm thinking people like the earlier ones as well. All right, well, thank you everyone for being here today. I really appreciate it. Again, make sure to fill out the form so you can get credit for being here. And yeah, have a great rest of your day. All right, bye everyone.